Thank you. So I'd like to welcome to the stage Dr. Rhoel Dinglasan, who is Associate Professor of Infectious Disease and the Director of the CDC Southern Regional Centre of Excellence in Vector-Borne Diseases Gateway Program. And Dr. Dinglasan is going to tell us about some novel falciparum biomarkers.
Cheers. Thank you for the introduction. Good morning, everyone. And so I want to talk to you a little bit about work that we've been doing in my laboratory that's quite separate from the HRP2, HRP3 story. As a bit of a background, our interest is really in my labs on asymptomatic carriage of malaria parasites. And so we were pushing the envelope in what we could do as a biochemistry mass spec lab to figure out, are there other biomarkers that are not HRP2 or 3 or LDH or aldolase that we can actually use. So I'm going to take you through a little bit of a story. I also want you to keep in mind that I am not a person that was interested in clinical management. I'm not interested in a clinical setting. So a lot of our work is predicated on understanding basically the landscape epidemiology of malaria carriage in sub-Saharan Africa. So moving forward. All right. So I'm pretty sure many of you have seen this slide before, and the big arrow down in the bottom left is to basically remind you that we're after something that is less than 1 parasite per microliter really estimate. So we wanted to push this idea, can we already develop a prototype rapid test based on less than 1 parasite per microliter? Can we go below microscopy just from the get-go? You know, so if we're going to fail, we're going to fail big. If we're going to succeed, potentially we succeed big as well. So this is my version of how I understand our capability of detecting malaria parasites in the field. So essentially, what we have up here is the pre-symptomatic individuals that, unless you want to do biopsy in liver, it's not going to be possible. What we're able to do with existing RDTs and microscopy as a gold standard. And down here in the bottom is asymptomatic carriage, which is a bit of both asexual parasite carriage as well as gametocyte carriage, and it's really we're at the level of molecular diagnostics, so which Jonathan and Tom had mentioned already. This is an old slide, but it's just again to follow up on what Tom Wellens had talked about, is that the FIND WHO study, there was a lot of failure initially in terms of sensitivity. Seeing that data, the Gates Foundation has funded the development of a 10-fold more sensitive HRP2 RDT. And so basically that, you know, we're at the level of about 10 to 20 parasites per microliter, which is really great. But from an individual who works in the field, I wondered whether or not we can stop using blood. Can we stop, you know, pricking the kids? I have kids too, and they hate it. They hate going to the doctor, right? And so one of the things I said was, well, what can we actually survey very quickly as a biofluid from individuals? And instead of blood, I started thinking about teaching myself a little bit about other biofluids. Here's another, sorry. How did it turn back? Okay. So here's another slide that basically highlights this idea that, okay, is PCR our best approach? Okay, when we talk about submicroscopic capabilities, must we go down the road of PCR? I wanted something, essentially, can we use it in a school setting, in a pharmacy, in the town meeting, what have you? You know, can we get out of the clinic? Can we get out of a PCR-centric approach? And then, of course, I only pulled out one example, but Jane already told you about all the other stuff that's coming out, and especially the stuff that you're learning about here in TropMed. Okay, so this is a target product profile that I imagined. This is a wish list that I wanted to try and achieve in my group. So the first one is, can we target a non-parasite, HRP2 biomarker? Now, as a transmission biologist in malaria, I was really interested in gametocytes. So I was looking for gametocyte biomarkers, but ideally, I really want something that's actually shared between asexuals and gametocytes. So if we are going to target gametocytes, I'm going to go for the more abundant gametocyte stage would be the female gametocyte, which has a 4-to-1 ratio, so 4 female gametocytes per 1 male gametocyte, which would be present in the blood. It must be essential for parasite development, so there's no mutation. It must be present across all Plasmodium species that infect humans. So what was the non-invasive biofluid? So, well, I wanted to tell you about saliva, and I'll get to that in a bit. must be able to, and Tom Wellens mentioned, can we make an antibody to this? Because I wanted to use a lateral flow immunoassay approach. Why? Because it's very familiar to all of you already in the field. It's familiar to the caregivers in the field. It's familiar to a lot of people. And lastly, can we use this for epidemiological landscaping for malaria? Okay, again, not trying to be I'm not a physician, so I don't have this clinical management paradigm in my head. So why saliva? Well, all of you will spit very freely from me. There's no needle prick, there's nothing along those lines. And if, for many of you, I mean, you guys, everyone spits. Just walking around the streets in Baltimore, everyone's just spitting. But why else saliva? So one of the things I did when I was here at Hopkins was I went to the dental school in Maryland, and I asked the departmental chairs, I go, teach me about dentistry, periodentistry, and all this stuff. Teach me about the mouth. And I got some old textbooks, and these are capillary loops shown here. Oops, sorry. These are capillary loops shown here of what's happening actually in the gums in a rat, okay? And it's actually also similarly conserved in humans. So we have a lot of capillary loops in our mouth space, essentially. And some old work from Bob Sinden, that talked about, where do these gametocytes, again, I'm very gametocyte-centered, where would they sequester? And they would normally sequester, they said, in capillary beds. So where are these capillary beds? They could be anywhere, but even in the pregnant woman, they're not even in the placenta. So I said, okay, fine. I hypothesized that gametocytes and other parasites would sequester in capillary beds, potentially in the mouth. So why, again, saliva? So in your gums, so gingivitis is my friend in this case. So gingivitis, basically, for those of you who have a dental background, results in a thinning of the barrier between what's actually present in our blood and our serum and what gets into our saliva. Okay, so it's essentially when you have a thinner barrier, you have more transudates that come from your serum and blood into your saliva, and they end up in your saliva, which is predominantly made out of water. So our hypothesis is that parasite proteins that are released following cell lysis or through natural release of, for example, microvesicles, make it into the serum transudate that ends up in your saliva. We've never seen parasites in saliva. It's a hypotonic solution. We don't actually expect a parasite to retain its features as an intracellular parasite. So we have never seen anything to date, but we also can't see by eye proteins extruding from the serum into the transudate. So our working hypothesis, again, is this. Gametocytes or other parasite stages sequester in capillary beds in the mouth, release exosomes and or microvesicles containing parasite biomarkers, and then make it into the saliva. So this is our experimental workflow. So we basically developed a highly sensitive two-dimensional liquid chromatography mass spec approach that also combines in-gel digestion. So we combine multiple approaches to give a better proteome. We spent a lot of time understanding saliva sample collection and stabilization in the field to bring it back to the United States for analysis. We used two different mass spectrometers, one for discovery, which is a QTOF, and one for absolute quantification using the triple quadrupole. And so we went out and they said, look, can we do it? So we first developed a reference pooled sample of the biofluid. So these are going to be from children in the field. And then what we did was, so if you have individual samples, you can take a little bit of each sample, make a pooled reference, and that allows you to do some discovery, and then go back to the individual samples that from where that pool was derived, and then do an analysis. Are these biomarkers actually present? So these are the 12 children that we worked with, and this is from Cameroon. And I share this with you because there are a few parasitemic parameters that we find very unique in Cameroon that many of you probably don't see on a regular basis. And that is actually shown here and highlighted, that there are actually sometimes more gametocytes than there are trophozoites. And these are all blood estimates. And also, we also took the opportunity here to understand, well, are people who are trophozoite positive, do they even transmit to mosquitoes? And the answer is no. And so again, just to give you a context here, I wanted to understand transmission. I wanted to look at people who are asymptomatic with subclinical and sometimes submicroscopic infections. So using these 12 samples, we were able to discover in the saliva of children 35 parasite proteins. And please don't get mad at me. It's under review right now, so I can't share with you all 35 of them. But I'll give you the summary here. So 35 proteins with less than 1% false discovery rate. So that's pretty much as good as you can get in mass spec. 2 peptides identifying each of these proteins. Of these, 19 of the 35 were found to be most abundant by relative spectral counts. Of these 19, 10 were gametocyte specific, and 7 were specific to asexuals. And of these 19, 2 were shared between asexuals and gametocytes. Okay, so that's what we have working forward. This is a bit from our published paper when we were trying to figure out, well, what are the parasite biomarkers? And why do I show this to you? Because when we were trying to come up with a male and female gametocyte proteome, which we published a couple of years ago, we basically generated a lot of reagents. And I wanted to see, well, okay, we discovered basically all these male and female gametocyte proteins. And from this list of 19, we essentially found this one. That's kind of serendipitous, if you ask me. And so we were really lucky in that regard. So this is a gametocyte-specific protein. And what you see here basically are synchronized gametocytes and trophozoites from NF54 and DD2 gametocytes and trophozoites. This is a Western blot. Interestingly, we find something in DD2. And we're not quite sure what it is. But at least for NF54, with an African parasite isolate, we find that gametocytes are positive for this protein. However, trophozoites are not. This actually goes against what's present in plasmid-DB. And so we have to actually question, what is in plasmid-DB? Are these highly synchronized stages that people have decided to do a full proteome on? And so, there's a lot of things that we still do not understand. At least in our hands, in our NF54, this protein, 121-8800, is present only in gametocytes. Okay. So what is 121-8800? So if you look it up, it's called Plasmodium secreted O-kinase protein 17. So that means that when it was first discovered, it wasn't even in gametocytes. It was in O-kinase proteome. And so these are all the different parameters for it. It's conserved across all plasmodia. Keep that in mind. It has mass spec expression that's higher in O-kinase. It's essential for asexual blood stages. Again, this meeting my target product profile. It's activated in gametocytes as a transcript. And its cell surface expression has been also described. It's not supposed to be expressed in trophozoites, at least in our hands. But again, there's a lower molecular band that we found in DD2. I didn't like the name, so we're going to call it something else. Instead of a secreted O-kinase protein, we call it PSSP17 as a sexual stage protein to basically capture the fact that we found it in gametocytes as well as in O-kinase. So we went back to our experimental workflow, and I said, let's push it. Now that we have a protein antigen that we found to be abundant in saliva of children, can we make that lateral flow assay? Okay, especially since I had all the reagents for this PSSP17 in terms of monoclonal antibodies. So we went back to the field, and again, to test my target product profile hypothesis, can we sample children, asymptomatic children in school? So by definition, they're not sick. And so we went through several schools in Cameroon, around the Mafu area, right outside of Yaoundé. We also tried a different approach. Can we go from house to house? And so this is in Enchelenge in Zambia, which basically is right across the border from the DRC. And so here we're showing different, we can collect saliva from children between 5 and 15 years old. This child's actually just a smaller version of a 5-year-old. And so what were we able to do? So again, we collected saliva, and this is a lateral flow test. So this is actually quite different. So we didn't have the money to make up and stripe all of our lateral flows. So we use a different method. So this is a lateral flow that basically has streptavidin as a capture on the test line. And then on the control line, you actually have anti-mouse IgG. So again, these are monoclonal antibodies against PSSP17. You mix these all together in solution with your biofluid. Okay, so it's saliva plus both antibodies, capture and detection, and then you let it go up the lateral flow strip, and they will either come to the treatment line or the control line. And what are these? These are recombinant PSSP17 that we use as our target analyte as we are developing this assay, and it goes down from 50 to 0.1 nanograms per milliliter. You don't see anything at 0.1 nanograms per milliliter. Mind you, we're only using 10 microliters of biofluid, okay? So we were pushing it. We said, although we could collect 1 to 5 ml of spit, we didn't want to actually use that. So we're only using 10 microliters. So here this is recombinant protein spiked into naive human saliva, and we use this as our test to move forward. Can we now do this in the field? And the answer is yes. So what we have on the positive control, again, is our recombinant protein, and you can see that on the top part of the panel C. Our negative control is we actually threw in HRP2, recombinant HRP2 in high amounts, just to see if we can screw up the biochemistry of our lateral flow. Okay, we've also done negative controls from everyone in my lab spitting into a cup, and so far they don't have malaria. That's good. We also did technical replicates. So A048 and A048B is the same child that we sampled in the same day, once in the morning and then once after she had finished basically doing her coloring book. I wanted to find out is it all about capture of timing? Like, you know, if I see you in the morning, I capture my biomarker. If I see you before lunch, it's gone. That's not a good biomarker. And then all the other case codes down from B357 down to D492. Now what's on the right? So MRM is the multiple reaction monitoring mass spectrometry approach. So essentially, PSSP17, we found a diagnostic peptide that we can actually identify using the triple quadrupole method, meaning to say we don't need much of your material. Okay, we're talking about less than about 20 microliters were used from each individual. So if it's MRM negative, so that's mass spec negative. If it's microscopy positive, well, that's obvious. And then we also did qRT-PCR. Now this is a gametocyte protein, and so we went after a gametocyte marker, PFS25. So in all of these children, and we sampled 364 children from both countries. Okay, we have matched blood that was preserved in RNA later, and we did an extraction for PFS25. Okay, so if it's qRT-PCR negative, that means we failed to actually amplify this gametocyte transcript marker from blood. Okay, so what do we have here? Essentially, as you go down the line, and for those of you in the back, if you don't believe the bottom ones, I don't blame you. I don't either. Okay, so we are now trying to optimize our lateral flow immunoassay, and let's see, who's in the back there? Can you see C022? Can you see the test line? Can you raise your hand if you can see it? Oh, thank you so much. So that one is qRT-PCR positive. It's less than 1 parasite per microliter in blood equivalent for the gametocyte. That is something that Tom Wellams is talking about. Can we get below 1 parasite per microliter? Because I wanted to see, can we make a lateral flow immunoassay that we are actually approaching the level of PCR? Now let me take a step back. You're wondering, what are all these images? It looks like a fluorescence assay, right? This was taken with the worst iPhone in my laboratory. Okay, it's like a 4. What are we up to now, 10? Okay, and we tried it with different phones. We tried a Nokia. We tried Huawei. I've never seen that, but I've seen it in the field. It's all over Africa. It's a Chinese cell phone company. And we said, can we just take images of our lateral flow? All right, so that's what we see here. So why is it fluorescent? So we are using as our detector, our bead detector, is no longer colloidal gold. It's a lanthanide element. Okay, it's called europium. So it's a europium chelate. And if you activate it with UV, it has a broad Stokes shift. You can actually see it in this room under this light or under a tree outside. Okay? And your camera, with a little bit of an orange filter that all of you can buy as like in sheets now, you can actually take an image of this lateral flow, and you can take all 10. Okay? So we tested it further. We said, is this secreted in a gametocyte culture? So these are axenic culture supernatants, okay, from stage 5 gametocytes, asexuals, and these are all synchronized, and as well as media control. And what you see here, it's in the supernatant. It is something that is actually secreted by gametocytes into the supernatant. Well, what's the difference between A and B is this. We actually ran this through a 22 micron filter. For those of you who do exosome biology, you know where I'm going. I believe this might be a vesicle-derived biomarker. If you don't have this filter, it also works, of course. Okay? We went a little bit further. I can only show you 2 here. But we actually went back to a different country, Sierra Leone, and we have 35 clinical samples because we were challenged. We go, well, you know, what use is this? You know, what's the utility of such a test if it doesn't work in the clinic? So we asked that. So these are symptomatic individuals. So what is the mantra? If you are symptomatic, you have a lot of asexual parasites, and 2 to 3% of that parasitemia, that parasite biomass, is gametocyte. And therefore, we anticipated being able to see it, and we did. For those QRT-PCR negative samples, they're positive. You know, we resample the saliva that we have in the freezer. And by the way, that's another advantage here. You can freeze the saliva, thaw it, repeatedly test it. It's very stable. That means it's not a free-floating protein. It must be in a vesicle. I'm telling that to myself. We don't have all the data for that just yet. Okay, so what are the pros? Identify submicroscopic carriers. We're approaching the level of QRT-PCR, at least for gametocytes. We use only 10 microliters of saliva. It's rapid. So if you have a lot of parasites, it's 3 minutes. If you don't, about 30 minutes. The studies were done blinded. We collected all the saliva and we just analyzed it blindly and then unblinded ourselves to go, oh, look, it worked. Because we wanted to remove bias. It's not HRP2. That's the first thing. What are limitations? We have no direct correlate in saliva. We have to go to blood and say, like, okay, well, how many gametocytes were in the blood? And that's not a very good test. So this is a major limitation for us. So right now, we don't know our true limit of detection. Me telling you that we can get below 1 parasite per microliter is fine, but that's not our collection design. We need to actually have a better experimental design to get the true sensitivity and specificity. So I apologize that I don't have that information today. What did I fail in our TPP? Well, we think it's important, right? But we haven't actually knocked it down ourselves. In Berghii, it seems to be very important for asexual blood stages. And Plasmodium species. So now we're working with our collaborators in Manaus, Brazil, to get Vivax samples and to see if it works with Vivax. And we're excited about that, and hopefully that we can get that information soon. I wanted to use this for epidemiological studies, and hopefully you would agree that it might be helpful to rapidly screen large populations without the requirement of physicians or healthcare workers. So a graduate student can do it, okay? And then also allows you to maybe help design your site selection in the field. Okay. All right, and lastly, for those who work in transmission blocking vaccines, hopefully you can make us design these a little bit better. All right, so I'm out of time. Hopefully I gave you a little bit of a flavor of a point of need test as a point of care test, so not to take it out of the clinic. Hopefully, we're going to get more biology information down at the bottom in terms of are these microvesicles or exosomes. And lots of people to thank, especially the kids and their moms and dads who gave us informed consent to spit in a cup, basically. So thank you very much.
Thank you, Rhoel. May I ask all the speakers to be on the stage for a moderated session? Anybody would like to ask the questions, please come to the microphone, and we'll go with the... Yeah, you can ask the question to any of the speakers or any other comments you may have related to the discussion of this session. Thank you.
Thank you. Good morning. So Jane invited me to this session this morning, and I told her that she would regret it. I'm Steve Harvey from Johns Hopkins University. I'm not a physician. I'm not an epidemiologist. I'm a social scientist, and I've spent a lot of time working on promoting rapid diagnostic tests in both the public and the private sector. And I find these results on HRP2 gene deletion, HRP2, HRP3 gene deletion very troubling. And I find them troubling from the perspective of having worked with a lot of providers, both at the community level in the public sector and at the health facility level in the private sector. So I think the advice that Jane has laid out here in terms of looking at the prevalence of gene deletion and what it means for false negatives as a result of gene deletion versus a result of other kinds of things like mispreparation of the test, which is something we've seen a lot of, makes a lot of sense from a programmatic standpoint, makes a lot of sense from the standpoint of WHO with a vision of what's happening all over the world, but it doesn't make much sense from the standpoint of what's happening between the patient and the provider. When we're promoting these tests to providers, what we're saying to them is you have to have a parasite-based diagnosis, you have to have a way of ensuring that what you think is malaria really is malaria, and for years now we've been saying to providers, don't use your own judgment about this. The issue's been providers will use the test, it'll come back negative, and the provider will say, well, I know what malaria is. I've been diagnosing malaria for years, and this test may be negative, but I know malaria when I see it, and this is malaria. And we're in the position of saying, no, you shouldn't be doing that. You should be respecting the results of the test and treating based on the results of the test. So now we have a situation where a lot of providers are saying to us, the test is always negative, and I have people that I'm sure have malaria, the test is negative, we're not treating them, what do I do about it? And our advice to them, if we base it on what WHO is recommending right now, is something that might take 2 or 3 or 6 months or a year to get a response, which is not going to be very helpful for the patient, for the febrile patient, particularly if that febrile patient is a child under 5. And I guess this leaves me with a real dilemma about what to say to providers and also say to the population, because while I understand Jane's point that we don't want to undermine people's confidence in the test, we do have a lot of providers saying the test is always negative. I think these people have malaria. We're saying to parents and caregivers, if your child has a fever, bring your child to a clinic within 24 hours because within 24 hours it can progress to cerebral malaria and death. But then we're saying on the basis of WHO guidance, if the test is negative, send them for microscopy when the child and their caregiver may not have access to microscopy, or compare HRP2 with PLDH when most programs won't have tests with both antigens. And so I don't know what the answer is. I'd be interested in any of your comments on it. Jane and I talked about it a little bit yesterday, but I think this presents a real dilemma and a real problem for the clinical interchange between the patient and the provider. Thanks.
Yeah, there's a lot of people lined up with questions, so I'm going to try to be brief. I mean, thanks, Steve, for highlighting again. I think the very difficult issue that we're facing, both I think as providers and users of RDTs, as well as at the programmatic level. So I don't think there's an easy answer. I think it's just that we don't want we need to get out in front of this, and that's what I'm saying. I think the best approach is to implement these surveys so that we can get representative samples and we can confidently tell the program, the population, whether or not we think that HRP2 deletions is having a clinical impact in the setting where they're working and seeing patients. And I think that strengthening a complaint system, strengthening such that, you know, at the level, even at supervisor, there is the awareness of this as a potential issue, and not to ignore complaints from providers of consistently negative results, to be able to bring those complaints up the chain. And I think one of the other things is arming programs with some of these other RDTs, such that they have a small stockpile of them and they can be used to do short investigations to address some some of those complaints, but there is no simple solution. I completely recognize that operationalizing this at the patient-provider interface is very difficult, and I think the best approach is surveillance to figure out if overall this is a problem, and if it is, where, and then adapt the policy accordingly.
Thanks, Ian. Michael, I had a question about the salivary biomarker. I think it's really exciting developments. But as a pediatrician, as a dad, I'm just wondering in young kids how you're going to, practically speaking, get samples, whether you've tried swabs or what material you're using, if that could impact your findings.
Yeah, so thanks for the question. In just time, I actually took out a couple of slides. We work with a company called Oasis Diagnostics in Seattle, and we actually have a pacifier. And we need 10 microliters. So when we first started doing this, we're like going, do we really need 5 ml of saliva? Thankfully, we don't. And so there's actually ways to collect it. It's not skin cells or cheek cells. You actually, it's a little bit of, it looks like a cigarette filter, for lack of a better description, and you put it aside the tongue, and it really sops up the saliva really rapidly. For the older kids, it looks like a lollipop, and you just put it aside, and they don't even notice it's there. And the adults, it's just a bigger lollipop. And so it's working really well. We've tested it with our tests. It doesn't remove the biomarker because of that. And yeah, it's really rapid. And I think the Gates Foundation is funding them for additional collection vessels.
I'm Deusi Shengoma from the National Institute for Medical Research in Tanzania. I have two questions, one to Jane and the second to Jonathan. Jane, as you can remember, last year when we were in Atlanta, we talked about the black spots in Africa, where actually we don't have data about HRP2, HRP3 deletion. And as you speak now, we just added only 2 countries, I think, Ethiopia and one more study which you mentioned. So it seems like over the 12 months, we just have done so much to be able to get more information from the different countries where we don't know what is happening. So my question is, as far as WHO has done quite a lot of work to harmonize the protocol, because we do have harmonized protocol for therapeutic efficacy studies and vector surveillance. And currently, actually, in Tanzania, we have included collection of samples for HRP2 deletion, whatever serological tests we can do. Is the WHO prepared to support this and spearhead surveys in many other different countries so that we can fill in the information and get more data from other countries where we don't have that information? And for Jonathan, I think as far as HRP2 and 3 end up in generating proteins, so I do feel that probably having serological tests could be tools for rapid screening a large population, and where we have kind of kind of results we can go in to confirm whether we have the deletion or not. But what do you say about the cross-reactivity between the HRP2, anti-HRP2 and HRP3 antigens, and how that could impact on serological surveillance? Thank you.
Okay, so I'll go first. So I would just say that definitely WH supports funding of these surveys. And if we had the funds, then we would support them. But at this point in time, we have limited resources to support surveys in countries. We can provide technical assistance. We're working to strengthen the international network of laboratories to conduct molecular analyses. Some of those laboratories have funding that they may be able to support analysis of samples coming from surveys. But I think it may initially end up being a bit of a patchwork. The Global Fund, I know that in some of the countries that are planning surveys, they're using funds from the Global Fund and also other leftover funds from other UN agencies. So I think that you won't have difficulty convincing your donors that this is a priority, but this is not going to be a really exclusively WHO sponsored effort. It will be really, countries need to think about how they will mobilize resources, and we at our side will encourage donors across the board really to support these kinds of efforts.
And thanks for your question about the HRP2, HRP3 cross-reactivity issue. From a molecular standpoint, at least in our current workflows, it's not a problem because we are focusing on exon 2 assays. However, I will say that depending on the assay you use, there are multiple ways around that. The most important thing is just to be very aware of your own assay's test characteristics. But I think the more important question has to do with serology and the cross-reactivity between these antigens. From my standpoint, this is actually a great thing because now there is some evidence, and the paper from Kenya that just came out today provides kind of a nice summary of that. I think Heidi Hopkins is here in the room. She's on that paper. The nice thing is if you lose HRP2 but you have HRP3, there is some growing evidence that you will still trigger a positive test. And so from a practical management standpoint, that's very good because with high parasitemia, you would still trigger a positive result and could justify treatment. But I will say that the testing in the lab for deletions is complicated by the similarity of the genes, and similarly, the antibodies, some of them or maybe all of them could bind both. But.
Thank you. Hi, Eric Rogier with the CDC. Thank you all for just really great presentations all around, really giving a comprehensive idea of the current situation that's faced. And so I have one suggestion and one question. The suggestion is something that's not seen in science a lot, is potentially reporting non-exciting results. And so I can imagine in this room how many freezers have thousands, if not tens of thousands of samples with DNA already isolated from a screening method that was not based on antigen detection but based on microscopy or some nucleic acid assay, and potentially how many manuscripts are even in this room that could report and fill in a lot of those blank areas on the map where we have no data on the HRP2 and 3 genotypes and from some of these areas. The question is, do you think that potentially sequencing HRP2 and 3 should become a standard reporting practice because we know of the variation of this protein globally and how that might actually impact, as been published on before, rapid diagnostic test results?
Well, I can take a stab at that. Eric, first of all, thanks for the question, and also I'm glad we could highlight your work on the Luminex platform, which I think is very useful for just this type of question. I have two thoughts. One is, yes, I agree with you. There's a lot of blank space on the map that we could fill in. However, from our own experience, I would caution against plunging into molecular assessments of this because, as I highlighted in my talk, the devil's in the details, and it can become a very challenging process. So I think perhaps the right approach would be to attempt to fill in the gaps, but maybe in a sequential way as suggested by Jane and the WHO, to prioritize where you do the surveys and perhaps do that in the context of WHO-supported lab network, which, as Jane's mentioned, is something that's underway now. But I don't know if you have anything to add, Kumar. You've thought a lot about this.
Yeah, I think we'll discuss with Eric after this. Anyway, all the network partners are meeting in the interest of time to help other people to answer the questions. We will go with the next question. Krishna?
I am Sri Krishna. I am a PhD student from National Institute for Research in Tribal Health, India. As Jonathan mentioned, there are different PCR conditions and reagents from different suppliers that may affect the results related to PF, HRP deleted or positive. So is it better, since it's idea to study the deletion of HRP2 in different areas around the world, in different countries, so is it better to calculate the PCR efficiency for a particular lab so that we can figure out what are the better PCR conditions to amplify this HRP2 gene, so that there are very less chances of mishappening related to PCR. I think as a person go for real-time PCR, first they calculate the PCR efficiency for quantification of any amplification, copy number of any gene.
So I apologize, I didn't catch all of that question, but I think your question was primarily related to how can we ensure high-quality PCR and standardized
PCR
methods.
condition. I am talking about the means insurity of the PCR condition. Those are best to amplify the PHRP2 gene.
Yeah, so I would say two things. First, in order to ensure high-quality PCR work in general, the WHO has sponsored an external quality assurance scheme, which Jane discussed yesterday, which for our own lab has actually been a very useful exercise to kind of standardize some of our own lab protocols. And then regarding standardizing testing methods, I think Chen Chang and Jane and others in this room have already started that process through their malaria journal paper in 2014, and there's an ongoing discussion about that now. I would say we don't have a conclusion to that, but hopefully at least the groups of labs who have worked on this in the past will be able to come up with some sort of more harmonized approach.
Thank you.
Okay, thank you. Vasant?
Hi, I'm Vasant Muralidharan from the University of Georgia, and I'm a basic biologist. We do sort of molecular genetics on falciparum, and thanks for inviting me here, Kumar. I had a question for Rhoel. He sort of mentioned that looking at essentiality of genes. I think going forward, it is vital that we ensure that these genes are essential. We have great gene editing tools now. We have whole genome screens for essentiality. And I hope I have the right number, Rhoel, but PFS 25 is not essential in Bergii. So it is vital that before we spend all our resources, we need to ensure that these genes are essential going forward.
Yes.
Thank you.
Oh, hello. I'm Christian Schneider from University of Mittweida, Germany. Great talks. Yeah, you really made up my morning. I have two questions. One is more like a comment to the second talk. So with the HRP2 deletions, you were talking a lot about prevalence. Now, from a theoretical standpoint out, is it really the right measure to look at? Because presumably you will only detect those, assuming you have perfect methods, if you have an infection that is exclusively infected with parasites having the deletion. So what's going to happen is, depending on transmission intensities, you will first underestimate the true frequency of deletions, especially in high transmission areas, and then all of a sudden you will highly underestimate. So if you look at the meets that started doing it that way, you will see all of a sudden a huge jump in prevalence of that, and say, oh, they appeared all of a sudden, but actually it really didn't. It is more like you should look at frequency maybe rather than prevalence. That's more a comment than a question. And my second question is regarding the saliva detection method. Great thing. My question is, I mean, how well really does it work for the different malaria kinds? For instance, in falciparum, it takes like a long time for gametocytes to form. So if it takes like 7 days already, I mean, isn't it cheaper to just use a fever thermometer?
Yeah. So one thing I didn't mention was I selected the specific domain on PSSP17 that is highly conserved across all plasmodiums that have been sequenced to date. It's about 87% amino acid level, so there's a good chance that the epitope is conserved. The second thing that you're asking, how well does it work? So one of the things that I'm hoping this lateral flow can do is actually detect developing gametocytes in the bone marrow. If you think
so
about
you will see it pretty much at the beginning.
Yeah,
of
because if this biomarker is exactly what we think it is, if it's secreted into the supernatant during development, then we could potentially see it, right? We don't need to actually see the parasite in the blood because, but it could get into the saliva.
Uh-huh.
that's what our hope is. I agree with you that, you know, if we have to wait for them to have immunocytes, then it's not good. But if you recall, the first thing I said was, this was a research project out of convenience. I had the reagents. This is not the biomarker that I actually want to sell.
Yeah.
Okay. We have one that's asexual and immunocyte conserved, and that's the one I really we'll hope we can get funding for, and that's really what I think is the best thing, because it'll address your concern. Like, do we have to wait for the immunocytes? No, we should go after trophozoites.
Great, thanks.
And I have one comment regarding transmission intensity. There's a paper that just came out by O.J. Watson, who's here in the room, that explores this a little bit from a modeling standpoint. That could be worth taking a look. It's an eLife from this year.
Thanks.
Thank you. Any of you have any comment? Tom, any of the speakers, any additional comment?
No?
Sure. It's a comment I'd like to take with Stephen offline, maybe after the session, discuss it some more. And that is, since the beginning, the idea that RDTs would be used, you know, to say there is no malaria, I found deeply unsettling as well. How do you prove a negative? And there, as we've come out in the session, there are many reasons an RDT will fail, many reasons PCR will fail. And as clinicians, I think we need to look at it as a case where the positives are in hand, and we do know then those are the cases. But when you have a negative test, then further evaluation is required. And you look at other tests for other diagnoses. I mean, there's a standard differential diagnosis of internal medicine. And then you need the certitude that you have made it, the case, the diagnosis correct. Now, in the field setting with malaria, where resources are limited and treatments are expensive, this becomes more difficult. It's a deep problem. We don't have easy answers. And I think going forward, it's our imperative as researchers and as clinicians to try to find new protocols, new ideas for the next generation tests and next generation treatments.
Jane, you have any more? No. If there is no comment, I thank all of my colleagues, the speakers, Michelle, but I would like to add one final comment. That is, you know, it is apparent from the discussion we had this morning, the most important diagnostic test is having a lot of challenges. So eventually we might lose this test. We are running out of time to have an alternate test. We had a great presentation from Rhoel, you know, what are the pathways for making new tests. But we need, as you know, it will take several years to bring it to the clinical or the field use. So at this point, you know, if you look at the product development for drugs or vaccine, there is a long-term vision from the malaria community and also a lot of investment. But we are nowhere near when it comes to the product profile for diagnostics. So there is a real urgency, and hope all the community and all the investigators here can help everybody come to the table and really address this as a real emergency, you know, to really control malaria. We have set up really high expectations in terms of elimination. Interestingly, that also came this year from WHO. Are we going to get there? Depends upon what kind of diagnostic test that's going to be available for us. I think with that note, I would like to close this session. Thanks again for all your patience and attention. Thanks to the speakers again for such a lively debate today. Thank you.